Hello everyone. Today I'm going to talk on the topic nucleic acid. The learning outcomes. After the session, the learners would be able to define nucleic acid, describe the biological function and composition of nucleic acid, and classify nucleic acids. Now, what is nucleic acid? Talking about it or the definition of nucleic acid, these are high molecular weight polymers of nucleotides containing a nitrogenous base, pentose sugar and phosphoric acid. This is a structure of ATP, that is adenine triphosphate, which is a nucleotide, which is an example of nucleotide where the nitrogenous base is adenine, pentose sugar is ribose, and you have 3-phosphoric acid that is attached in triphosphate bond. Let us pause the video and try to answer this question, that is, what are the various biological functions of nucleic acid? Once you are done with answering this question, play the video to check the answer. What are the functions of nucleic acid? So first and foremost, or the main function of nucleic acid is storage of genetic information. The information required for the entire body is stored in a sequence which is known as gene, and this gene is then responsible to give information for the synthesis of various other biomolecules. The second function is a replication or copying of the information or synthesis of more and more DNA. The process is called as replication. Then from the DNA or the information stored in the DNA, RNA is synthesized or the information is passed on to what is known as messenger RNA. This messenger RNA act as a information and is used for synthesis of various proteins. Here is a diagram which is going to explain the main important functions of the nucleic acid. The first one, you can see DNA is copied to give you daughter DNAs. The process is known as replication. From the DNA, the information stored in the DNA is then transcribed to form or to synthesize RNA. From the information which is now being copied from the DNA to RNA, mainly from the mRNA, it is translated to synthesize biomolecules called as proteins. That process is known as translation. So these are the main important function of the nucleic acid. The other functions also include, they are also the component of coenzymes. So if you talk about these nucleotides, that is ATP, these ATP also form a component of coenzymes like NAD, NADH, FAD, etc., which are involved in biochemical pathways. Coming to composition or what are the components of nucleic acid? So first one is nitrogenous base. The second component is pentose sugar, and the third component which is present in these nucleic acid are phosphoric acid. Coming to the first one, nitrogenous bases. These nitrogenous bases are nothing but derivatives of aromatic heterocyclic compounds. There are two types of these nitrogenous base depending upon the structure. These are purines and pyrimidine. Purine is two rings fused together, two heterocyclic rings fused together. These two heterocyclic rings are, one of the ring is a 6-membered ring which contains 2 nitrogen, which is called as, the ring is known as pyrimidine, and the other one, the 5-membered ring, which again contains 2 nitrogen, is imidazole. So purine structure is a fused ring of pyrimidine and imidazole. And pyrimidine, as we already talked about, it's a 6-membered ring containing 2 nitrogen. So these are the 2 types of nitrogenous bases which are present in nucleic acids. These can exist in keto or enol form depending upon the pH of the environment. Although the keto form is more predominant. Second component of the nucleic acid is pentose sugar. So you will find a pentose sugar in the structure of nucleic acid. This pentose sugar is nothing but ribose. Ribose is an aldose which consists of 4 carbon, 2 types or 2 types of ribose sugar which is found in nucleic acid, which is going to further differentiate the nucleic acid into DNA and RNA, is the ribose and the deoxyribose form of pentose sugar. So this is a structure of ribose and deoxyribose sugar. So what is the difference between these two structures? A ribose sugar has an hydroxy group attached to the 2' prime position, whereas deoxyribose sugar does not have an oxygen at the 2' prime position. That gives the name as deoxyribose sugar. Nucleosides and nucleotides. Before we go into the types of nucleic acid, we have to see these 2 terms, that is nucleosides and nucleotides. Nucleosides are basically made up of nitrogenous base and pentose sugar, whereas nucleotides are made up of nitrogenous base, pentose sugar and phosphoric acid. Now, what are the different types of nucleic acid? Now, as I told you before, as we saw the pentose sugar structure, there are these 2 forms of ribose sugar which is present, that is ribose and deoxyribose. This pentose sugar is a main criteria which differentiates these 2 nucleic acid, that is DNA and RNA. So DNA, or it is also known as the full form of DNA, is deoxyribose nucleic acid, or it is also known as deoxyribonucleotide, is made up of, or it is generally a polymer of deoxyribose nucleotide. So deoxynucleic acid would be the polymer of deoxyribonucleotides, whereas RNA, these are the polymer of ribonucleotides. So the polymer made from many of these deoxyribonucleotides, the nucleotides which are made up of deoxyribose, if they are joined together, they are going to form DNA, whereas ribonucleotides, where the sugar is ribose, those nucleotides, when they join to give you a polymer, that is known as RNA. Now RNA are further classified into 3 types, that is mRNA, that is messenger RNA, rRNA, that is ribosomal RNA, and tRNA, that is transfer RNA. These are named because of the different functions they have. So with this, we come to the end of the presentation. Thank you.